Hello, can somebody confirm that you guys can hear me?
Hello.
Thank you. Okay, before we begin, are there any questions? Today is February 11th, and we're supposed to be talking about Lab 7, throat and urine cultures. We'll also talk about your unknown, well, project a little bit. Any questions about any of that? Let me share my screen here. That's not right. Excuse me. All right. About the unknown project, I'll be sending you the first three test results tonight. It'll probably be after the 6:30 lab. Is that right? So sometime around 7:30, 8:30, be expecting your results for the first three tests. Any question about any of that? So take a look at the results and the table of the unknowns and figure out which next test to request and request it in your unknown notes. All right, lab 7, throat and urine cultures. There's some reading to do in this lab. So read page 392 about nose, throat, and mouth normal microbiota. There's some reading on the beta hemolytic streptococci, and that's in this lab module in the appendix. There's also a little discussion about Haemophilus, that family of bacteria, specifically Haemophilus influenzae, um, also in the appendix of this lab module. And then there's a page to read about the oxidase test, another page to read about the coagulase test, excuse me, the catalase test. And then there's a little bit to read about the coagulase test in this lab module. Any question about any of that? There is some video clips to see in this lab. There's no virtual lab. The learning objectives for this lab are be able to explain how to properly collect throat and urine specimens for routine culture in the lab, list the common bacterial pathogens isolated from throat and urine cultures, know the culture media used to cultivate these pathogens, recognize which tests are used to identify throat and urine pathogens, and then understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. We will be talking about the XV factor test, but you do not need to, I guess, really understand it or be able to explain it. Once specimens are received in the lab, they are logged and processed in a timely fashion. Specific culture media are inoculated and incubated for specific throat and urine specimens. Below is an overview of the culture media used to cultivate common bacterial pathogens isolated from throat and urine cultures. For the throat, we really only have two culture media for growing throat specimens. Number one is blood agar, and it is used really for everybody. Chocolate agar is used particularly when we're taking a specimen from the throat of a child or a baby, and Streptococcus pyogenes is the pathogen we grow on blood agar. But Haemophilus influenzae, it cannot grow on blood agar and has to be grown on chocolate agar. And Haemophilus influenzae is something you can obtain from the throat of children, particularly young children and infants. Any question about any of that? All right, for urine specimens, we grow them always on blood agar and then McConkey's agar or eosine methylene blue agar or EMB. So this one always on blood agar, but either on McConkey's agar or EMB. McConkey's agar and EMB, if you don't remember, grow gram negatives. And then we're looking for these common bacterial pathogens in the urine: Streptococcus, Enterococcus, Staphylococcus, and all of these grow on blood agar. And then Enterobacteriaceae, especially E. coli, but other Enterobacteriaceae and Pseudomonas, and they will grow on McConkey's agar and EMB as well as on blood agar. Any question about any of that? Sore throats may be caused by either viruses or bacteria. If you see white patches on inflamed tonsils or in the back of the throat, it is indicative of a bacterial infection. So a disease like that would be a bacterial infection. If it's just red, it's likely to be a viral infection. The most common isolated bacterial pathogen is Streptococcus pyogenes, also called Group A Streptococcus, or the abbreviation GAS. I use the word Streptococcus pyogenes to name the species, but you could go by the group antigen, which is kind of an older way of doing it. Strep throat is caused by Streptococcus pyogenes. And what you do is you simply swab the back of the throat or the tonsils where there's redness or white patches. You avoid touching the tongue, the sides of the mouth, and the roof. And then you swab that on blood agar. Patients who test positive for Streptococcus pyogenes are placed on antibiotic therapy. And that's because Streptococcus pyogenes has sorry, I'm drawing blank on the name of this, a glycocalyx, which we can see under the microscope. So we call it a capsule. And you grow Streptococcus pyogenes on blood agar because it is fastidious organism. It will not grow on general purpose media. It requires certain additives to be added to the media to grow, and the additives are supplied in the blood. And usually they use sheep blood with tryptic soy agar to get blood agar. And then you examine the plate for any small, clear colonies surrounded by a complete zone of hemolysis. That's what they mean by clear. The colonies are actually small white colonies. And you're looking for beta hemolytic colonies, which are produced by Streptococcus pyogenes or some other bacterium. Only 2 biochemical tests are required to definitively isolate Streptococcus pyogenes if it causes beta hemolysis on a blood agar. We run the catalase test and then the basotrace and susceptibility test. So here we have blood agar not showing complete hemolysis, and right here we have blood agar showing complete hemolysis. And as you can see, that little spot right there, the white spot is the bacteria, and then this clear region around the colony is beta hemolysis. And you can see beta hemolysis in these streaks, but the majority of the streaking is not beta hemolysis. And that's because the throat is home to many different bacteria species. So when you swab, you're going to get more than just Streptococcus pyogenes caused by somebody who has strep throat. And right there, this right here is gamma hemolysis, but over there, you can see some alpha hemolysis, the darkening of the blood. And right there around that one too, there's alpha hemolysis. Any question about any of that? All right. So like I stated, if you get beta hemolysis in blood agar, you only need to run 2 more tests to prove that it is caused by Streptococcus pyogenes. The first test is the catalase test. Catalase is an enzyme produced by most bacteria to break down hydrogen peroxide into oxygen. So here we have hydrogen peroxide. If you have catalase around, it'll break it into water and oxygen, and that production of oxygen will cause bubbling in the hydrogen peroxide. So you look for bubbling. Bacteria test positive for catalase production. Oxygen is observed, and there we have a positive catalase test, and that's a negative catalase test. Streptococcus family, as well as Enterococcus species, test negative for catalase production. Most bacteria that grow in the air are positive for catalase. So if it's negative, then it could be Streptococcus, but it could be another member of the family Streptococcus or Enterococcus. And then you do the basotracin susceptibility test. Basotracin is an antibiotic, and what you do is you smear the agar from the blood agar plate on another plate, and then put down the basotracin disc, and then incubate the plate upside down in the incubator, and take a look at it after growing it overnight or for 24 hours, something like that. And if you get a positive basotracin test where there is a zone of inhibition of a certain size, then it's positive for Streptococcus pyogenes, which here is labeled group A Streptococcus. If, on the other hand, it's negative, meaning there's no zone of inhibition, or the zone of inhibition is smaller than a certain size, then it's negative, obviously, in the basotracin test, and it's not Streptococcus pyogenes or group A strep. Any question about any of that? All right. Another organism that can be clinically significant when isolated from the throat is Haemophilus influenzae. It's a gram-negative coccobacillus. Haemophilus influenzae is one of the most common causes of ear, eye, and or sinus infections in babies and young children. Haemophilus influenzae is fastidious, but it will not grow on blood agar. So you need to grow it on chocolate agar, which is where the red blood cells of a blood agar plate have been added to the agar when the agar is first out of the um, what is that called? The autoclave. And so the agar is really hot, and that lyses the blood, making the agar look brown, which is why they call it chocolate brown, because it looks like chocolate. And that will release the chemicals and additives from the blood, which are required for Haemophilus influenzae. So if you streak Haemophilus influenzae on a chocolate agar plate, you will get the colonies growing. But if you were to streak it on blood agar, you would not, unless you were to do a specific test, and that is you can streak Staphylococcus aureus on the blood agar in a single streak going this way, and then streak the Haemophilus influenzae this way on the blood agar plate. And you'll note that the Haemophilus influenzae are growing around the Staphylococcus aureus. And right here we can see the Haemophilus influenzae, but up here, which has also been streaked, we cannot see Haemophilus influenzae growing. And that's because near Staphylococcus aureus, Haemophilus influenzae is growing because the Staphylococcus aureus is lysing the blood. And actually, you can see that if I blow it way up high. You can see the streak of Staphylococcus aureus right there. This white area around it is actually beta hemolysis that is releasing or lysing the blood, which will release the additives from the blood that are required by Haemophilus influenzae to grow. And that's why Haemophilus influenzae is only growing near the streak of Staphylococcus aureus. Any question about any of that? All right, so you don't really need to know that you could streak blood agar with Staphylococcus aureus and then streak the Haemophilus influenzae perpendicular to it. And then the Haemophilus influenzae will grow around it. But you do need to know that chocolate agar will allow you to grow Haemophilus influenzae. The factors that are required by Haemophilus influenzae, which are supplied by the chocolate agar, are the X and the V factors from the red blood cells. All right, any question about throat cultures or isolating throat cultures? If not, let's move on to urine cultures. Urine cultures are used to diagnose urinary tract infections associated with cystitis, urethritis, and polynephritis. Normally, urine is fairly sterile. If you recall, we recently learned within the last 10 or 15 years that urine is not sterile in the bladder of females. And to tell you the truth, I don't know about males because no one's ever done research on it, at least as far as I could tell. Females have a shorter urethra than males, and so they are more likely to get infectious bacteria growing up the urethra and then colonizing the bladder, and that makes females more likely to get a urinary tract infection. To collect a specimen from urine, you clean the genital area prior to collection and then use one of three methods to obtain urine. You can use the clean catch midstream, where the patient starts urinating, and in the middle of the urine stream, they collect the urine. This reduces the cultivation of transient organisms from the urethra. Does not remove them, but does reduce them. The second way you can collect the urine is by a catheter, sticking the catheter through the urethra into the bladder, and then sampling the urine directly from the bladder. But realize if you do that, that the catheter is going through the urethra, picking up microbes from the urethra, and then depositing those microbes in the urine in the bladder. And so you can pick up bacteria from the urethra, even when you put the catheter in the bladder. But it is cleaner than the clean catch midstream method. And then the third way, which is the most invasive, is suprapubic aspiration, where you stick a needle through the person's abdomen and into the bladder, and then sample the urine in the bladder. Obviously, patients don't like that procedure very much, and they're not really fond of the catheter being stuck in their bladder either. They would much rather do it from the clean catch midstream. Any question about how you sample urine? Once the specimen is received in the lab, you streak 0.001 milliliters, that's 1 microliter, of the urine onto a blood agar place. We have a special inoculation loop which actually measures the urine, so it will place only 1/1000th of a milliliter or 1 microliter onto the blood agar plate. And we get all of the organisms to grow on blood agar, including some fastidious organisms, excuse me. But we also streak the urine onto either McConkey's or eosine methylene blue or EMB plates, since the most common isolate causing a urinary tract infection is E. coli or another coliform. And they specifically grow on MAC or EMB because the coliforms are all gram-negative. And McConkey's and eosine methylene blue select for gram-negatives. They're called coliforms because these bacteria are found in the digestive tract, in the colon as part of the digestive tract. So there is the loop that actually transfers 0.001 or 1/1000th of a milliliter of urine onto the blood agar plate. And usually they streak it in a streak this way and then streak it that way. And then they take a sample using a regular loop and then streak it out, usually for colony isolates on the MAC or EMB plate, using the normal streaking method for isolating colonies. The interpretation for a positive urine culture is multifaceted. Some factors that are considered when you're looking at two plates from urine cultures is the type of collection. Was it voided, meaning the clean catch method, versus a catheterized specimen, obviously with or suprapubic aspiration. The gender of the patient, females have more bacteria from the urethra than males. So there'll be more contamination in the female urine. And then the number of colonies per mil of urine. The presence of microbial growth, polymicrobial growth, meaning colonies of many different types, indicate that there may have been poor collection technique of the patient. If they're doing clean catch midstream, they might not have done clean catch midstream. They may have collected at the start or the end of the urine stream. All right, so once you have the culture growing on either blood agar plates or McConkey's or EMB for a urinary culture, we then have a couple of tests to run to help isolate the species. You can run the coagulase test. Coagulase produces the enzyme, looks for the production of the enzyme coagulase, which is used to differentiate Staphylococcus aureus from other Staphylococcus species. Coagulase can contribute to the pathogenicity of Staphylococcus aureus by converting plasma fibrinogen into fibrin, which will form a clot around the bacteria. And the clot around the bacteria will make it more difficult for the host immune system to attack the bacteria. And that's one way the Staphylococcus aureus can survive in the patient when the Staphylococcus aureus has made the initial bacteria infection and the bacteria are low in numbers and susceptible to being removed by the host immune system. The Staphylococcus aureus create a blood clot, and that protects the Staphylococcus aureus, especially from phagocytosis by the white blood cells. They can't easily get through the clot to kill the bacteria. So in the coagulase test, we put plasma usually, and in the presence of the enzyme coagulase, which comes from a bacteria like Staphylococcus aureus, you get a fibrin clot. And there are two tests. You can use a slide test where there's plasma here, and this is a negative control where there's no clotting. And there the plasma has began clotting. You see clumping on the slide. But you can also use the tube test. And if the coagulase is present, the plasma will clot, shown here, will clump together. And the negative, the plasma will be a liquid, and you tilt the tube and you can see the plasma is running. Any questions about the coagulase test? All right, the most common bacteria isolated from a urine culture causing a UTI is actually E. coli. But other members of the family Enterobacteriaceae are coliforms which may also cause a UTI. And I think I mentioned that's why we tend to grow urinary tract infections on EMB or McConkey's because you can get the family Enterobacteriaceae to grow on those media. However, another isolate that can cause a urinary tract infection is Pseudomonas aeruginosa, which is another gram-negative bacillus causing UTIs. The Pseudomonas aeruginosa and all members of the Pseudomonas are aerobic and non-glucose fermenting, and they're oxidase positive, in which case the Enterobacteriaceae are all the reverse of that. They are gram-negative bacillus. They can cause a UTI, but the Enterobacteriaceae are facultative anaerobes. They're not aerobic. They all ferment the sugar glucose, and all of them, family Enterobacteriaceae, are oxidase negative. So the oxidase test is a test we run to determine is the bacteria Enterobacteriaceae or is it Pseudomonaceae? And I don't know why they do the oxidase test because they could do the glucose fermenting test or even aerobic respiration to determine that, but they tend to do the oxidase test. Oxidase is an enzyme that is looking for the enzyme cytochrome C oxidase, which is in the electron transport chain of an aerobe in the electron transport chain. So if the bacteria can perform aerobic respiration and have the electron transport chain, they will have cytochrome C oxidase, and they will be oxidase positive. If, on the other hand, they do not have this electron transport chain of aerobic respiration, they will be oxidase negative. So we use the oxidase test to differentiate between Pseudomonas aeruginosa and other members of the family Enterobacteriaceae because all the members of Pseudomonaceae are oxidase positive and the Enterobacteriaceae are oxidase negative. If on the oxidase test right here, if you see a purple or indigo color developing within 30 seconds, it is oxidase positive. If it doesn't happen within 30 seconds, it's oxidase negative. All right, any questions about the tests we use for looking at bacteria that have grown on blood agar or EMB or McConkey's for a throat culture, not a throat culture, a urinary tract infection culture? All right, if not, know the terms of this chapter and realize we're not going to be doing this lab because everything we do will be online. However, you do need to know how to perform them. You will be held responsible for that. And then we have a little video you can watch on running the catalase test. Click on that link. We have a little video you can watch on running the coagulase test. Click on that link. And then a little video you can watch on running the oxidase test. Click on that link. All right, let's go to the worksheet now. So the first question is for questions 1 to 3, identify the principle used to detect the presence of one of these three enzymes, the catalase enzyme, the coagulase enzyme, or the oxidase enzyme. So 1, which enzyme converts fibrinogen into fibrin? That's 1A. And then 1B, name a microbe that tests positive for this enzyme. 2, what enzyme detects the presence of cytochrome C proteins in the electron transport system? And then 2B, sorry, name 2 microbes that test negative for this enzyme. For 3A, which enzyme hydrolyzes hydrogen peroxide into the non-toxic products water and oxygen? And then 3B, name 2 microbes that test negative for this enzyme. And you don't need to say the species there, you can just say the family, but you could put the species if you wish. And then 4, which enzyme is used to differentiate between the following? So which enzyme do you do to determine if it's Staphylococcus from Streptococcus? Which enzyme do you use to determine is it Staphylococcus aureus from another Staphylococcus species? And then which enzyme do you use to differentiate between Enterobacteriaceae, like E. coli, and the Pseudomonas family, the Pseudomonaceae? 5. Concerning specimen collection for routine throat culture, what areas of the throat should be swabbed? And then 5b, what areas of the mouth should be avoided? 6. What is the most common species causing bacterial throat infections? So you must name the species. 7. What test is used by microbiologists to differentiate between beta hemolytic group A streptococcus, meaning Streptococcus pyogenes, from the other beta hemolytic streptococcus species isolated in a throat culture? 8. Answer the following concerning a child's throat culture. 8a, state the two non-selective agar plates that are inoculated, right here and right there. And if you go and use, if you go and use a AI to get your answer, it may be wrong because you have to list an answer that is talked about in this lab module. Any questions about any of that? I suppose if you were to list another media and put in the additive, I'll accept that. But if you don't have the additive, you've got to list the media that's mentioned in this lab module. All right. 8b, what organism can be isolated on CAP but not on BAP? 9, list two media that are used to grow urine cultures. 10, list 3 reasons why a urine specimen should be rejected for culture, and that is covered in the appendix to this lab module. Let me show you the appendix. I forgot to do that earlier. After the questions, which you might not even have, there's the appendix talking about gram-positive streptococcus, specifically talking about Streptococcus pyogenes. And then, no, never mind, that's not something. Talking about the beta hemolytic streptococcus, which Streptococcus pyogenes is, but there are others, streptococcus, which are also beta hemolytic. And then we're talking about Streptococcus pyogenes. And then we're talking about the gram-negative bacteria, specifically the members of the Haemophilus, talking specifically about Haemophilus influenzae. And then lastly, we're talking about the coagulase test, and then urine cultures. All right. So question number 11, how much urine is placed onto the surface of the blood agar plate? And 12, what is the most common bacteria isolated from patients with a bladder infection? Once again, you must name the species. And then 13, identify the gram reaction, what's the gram stain, and the cell morphology, the shape, not the growth arrangement of these bacteria. A, Pseudomonas aeruginosa, B, E. coli, C. Staphylococcus aureus, D. Streptococcus pyogenes, and E. Haemophilus influenzae. All right, any questions in this lab? All right, I'll be here until 4 o'clock to answer any questions, or I'll be here until the last student logs out. If you do get help from another student and you take their answer, you should put it in quotes where you got it. or um, at least write it in your own words. And if you're getting help from somebody, make sure they know what they're doing before you take their answer. Any questions about any of that? All right, go ahead and get started on the lab.